Hello and welcome. My name is Vincent Delle. I am a veterinarian by training, and I have the privilege of having the charge of the scientific secretariat of the European Q Fever Committee. I welcome you to our first live webinar. Today we are dealing with practical aspects of Q fever in small ruminants in Europe, and we have three speakers with 15 minutes talk for each of one of them, and this will be followed by a 15 minutes session of questions and answers. So please write your questions in the chat section, and I will go through them after reading them at the end of the third presentation. And without further waiting, I give the floor to George Valiakos. George is Associate Professor of Bacteriology and Bacterial Diseases in the University of Thessaly here in Greece, where we broadcast from. And he will talk about the clinical science and epidemiology of Q fever in small ruminants. And George, the floor is yours.
Thank you, Vincent. Good afternoon, everyone. So, Q fever is one of the most complex and economically devastating zoonotic diseases currently circulating globally. Historically, coccidiosis in livestock has been underdiagnosed, thought to be a background finding or underestimated by more, let's say, aggressive pathogens such as Brucella, Chlamydia abortus, or Campylobacter. However, the huge scale of the Q fever outbreak epidemic in the Netherlands between 2007 and 2010 caused a shift in both veterinary pathology and public health awareness. In this first presentation, we will focus on describing the specific to small ruminants clinical signs of the disease and the main epidemiological characteristics. However, to fully understand Coxiella, it is important to first examine its special macrobiological structure. Coxiella is a very small, obligate intracellular bacterium with a tropism to macrophages and placental trophoblasts. It is pleomorphic, exhibiting a complex biological cycle, altering between distinct forms and adapting to both the internal of the host's immune cells, like the macrophages, and the external conditions of the physical environment. The pathogen exists mainly in two distinct forms, the small cell variant and the large cell variant. The pathogenesis begins when the small cell variant is entering the host macrophages or the trophoblast cells of the placenta. Rather than being destroyed, Coxiella adapts, and within the acidic environment of the phagosome, the bacterium is activated and transforms into the active large cell variant, which rapidly multiplies. When the host cell is exhausted, when the host cell undergoes lysis, the large variants change into the small variants before being expelled into the environment. This form functions as a pseudospore and is the main one that survives in the environment and infects a new host. The structure of the pseudospore is characterized by extraordinary environmental resistance, being able to persist in conditions that would eradicate other bacteria. The pseudospore is highly resistant to extreme desiccation, changing environmental temperatures, high levels of UV radiation from sunlight, and many standard chemical disinfectants. Studies have demonstrated that the pseudospore can remain fully viable and infectious in contaminated soil and dust for up to 4 months, in meat for 1 month, in wool for 10 months, in animal feces for 2 to 3 weeks. While the pseudospore can withstand direct exposure to heat at 60 degrees for 30 minutes, it is inactivated by high temperature commercial pasteurization, so dairy processing is critically important for reducing foodborne transmission risks to humans. After this brief introduction to the pathogen, let's see some details on the pathogenesis and clinical signs that characterize the disease in small ruminants. Trophoblasts in the allantohorion of the placenta are the primary target cells for Coxiella. Upon colonization of the reproductive tract, the bacterium uses the nutrient-rich, immunosuppressed environment of the placenta to replicate to huge densities. During parturition, or in the event of an abortion, the materials can contain up to 1 billion bacterial cells per gram. In the image, we can see on the left that the allantohorion of an abortion case in an infected goat is severely thickened with a yellow, brownish exudate, comparing to that of a non-infected goat on the right of the image, which is thin, glistening, and transparent. while the cotyledons show no obvious macroscopic changes. The last probably explains the birth of many healthy animals, even if the mother is infected and shedding the bacterium, as the cotyledon villi are the main involved in the exchange of gases and nutrients between the mother and the kid. In both humans and animals, the majority of Coxiella infections are subclinical and asymptomatic. However, when clinical disease occurs in small ruminants, it is characterized by acute reproductive failure. Clinical cocciellosis in small ruminants is characterized by the so-called APSW complex, an acronym for the words abortions, premature births, stillbirths, and weak offspring. Late gestation abortion is the most prominent and serious symptom observed in infected farms. In small ruminants, Coxiella may cause sudden abortion storms, which is much less common in large ruminants and cattle, where the disease often takes a more chronic path. A primary infection occurring prior to conception or during early gestation gives the pathogen enough time to colonize the placenta, leading to late-term fetal abortion, premature delivery, stillbirths, or even birth of weak offspring. Now, regarding the role of Coxiella in secondary reproductive disorders, such as retained placenta, health fertility issues due to returns to estrus, endometritis, or mastitis, it remains a subject of ongoing research. In small ruminants, clinical signs outside of the APSW complex are underreported, assuming that sheep and goats may not suffer from this. Recent studies, particularly emerging from goat dairies, have begun to challenge this assumption. Reports from the Netherlands indicate that a significant number of aborting goats subsequently developed clinical metritis, accompanied by a spike in perinatal mortality among seemingly healthy kids born in the same contaminated environment. Further studies are needed to measure the impact of the infection on the frequency of retained placenta and fertility issues, or even mastitis. Goats are recognized as being significantly more sensitive to Coxiella than sheep. In caprine herds, an initial outbreak can be catastrophic, with abortion rates occasionally impacting up to 50 to 90% of the pregnant does within a single kidding season. It is important here to refer to another clinical sign that was recently reported to be connected with high Coxiella sender goats, a reduced milk yield, about 17%, in comparison with low sender goats, a loss of almost half a liter daily, which is really concerning for the impact that this bacterium may have in the productivity of dairy farms. To sum up, in the event of clinical signs of the APSW complex, and especially late gestation abortions, Q fever should always be suspected. Moreover, it is generally accepted that in healthy flocks, the baseline of visible abortions typically is below 2%, so an abortion rate exceeding 2% is indicative of an endemic disease present, requiring close monitoring. However, abortion levels exceeding 5%, or a sudden temporal or spatial clustering of abortion, such as, for example, multiple events concentrated within a 1 to 2 week window or localized to a specific birthing pen, requires an immediate diagnostic investigation for Q fever in your differential diagnosis. Now let's see some info regarding the epidemiology of the disease, focusing on the prevalence and transmission pathways. Seroprevalence studies across various European countries have demonstrated a high degree of exposure of small ruminants to Coxiella, reaching rates from 25% to almost 60% in animal level, and much higher, up to 100% in farmer level. This discrepancy between animal and herd level demonstrates the complex infection dynamics of Coxiella. The pseudospore is so ubiquitous that almost every single herd will eventually encounter the bacterium, resulting in this near universal herd level seropositivity over time. However, within a herd, individual animals will demonstrate various immune responses. They might either rapidly clear the infection, restrict it into tissue macrophages, or actively shed the pathogen during specific windows, such as parturition and abortion or concurrent illness. Therefore, a one-point-in-time individual screening will always underestimate the true impact of the pathogen. Attempting eradication through reactive means, like removing or cooling sick animals, alone is practically impossible without addressing at the same time the continuous environmental persistence of the pathogen. While the biological host range of Coxiella is extraordinarily broad, the domestic ruminants are acknowledged as the primary epidemiological reservoir, being responsible for the environmental contamination and the human zoonotic spillover. Infected small ruminants shed the bacteria heavily through multiple biological routes, including milk, feces, urine, and most importantly, genital secretions. The most biologically dangerous and significant event is parturition. The expulsion of infected placenta and amniotic fluids releases a huge load of the pathogen directly into the immediate farm environment. Transmission between the animals occurs via the airways and the inhalation of contaminated aerosols. Research indicates that goats shed significantly higher bacterial loads through vaginal mucus than sheep during the first months postpartum. Furthermore, caprine species demonstrate a significantly higher shedding via the rectal route and for a much longer continuous duration, heavily contaminating the farm compared to vine species. The pathogen shedding is not a single season event. Multi-year monitoring of infected goat herds over 7 consecutive killing seasons revealed deep persistence. While the extracted bacterial load decreased significantly 1 year after the initial outbreak of abortion storms, a high percentage of animals continued to shed the pathogen subclinically, and it was not until the 7th killing season that active shedders were completely undetectable via PCR. This multi-season shedding, as we understand, cancels any short-term reactive veterinary control measures. If a farmer culls aborting animals but fails to implement long-term herd-wide vaccination or environmental decontamination, the subclinically infected replacement animals will simply reignite the outbreak during the subsequent birthing cycles, continuing the productivity loss. Once shed in the external environment, the pseudospores are incorporated into farm dust. Because the infection dose is exceptionally low, the infection dose to cause disease in humans and naive animals may be fewer than 10 bacteria. Aerosolization is the primary driver for massive multi-regional outbreaks. The pathogen's capacity for wet-borne dissemination is high and represents a big challenge for public health authorities. Studies have indicated that farms located in areas characterized by flat, open land shapes, high animal densities, elevated ambient temperatures, and high wind speeds face a high risk of infection. On the other side, annual precipitation has been identified as a protective factor. Heavy rainfall seems to act as a natural dampener, suppressing the formation of dust and preventing the aerosolization of the pseudospores. So, due to this high environmental persistence, it is necessary for the farmers to implement measures that will aim at preventing the introduction or reintroduction of the pathogen, protect animals from productivity loss due to reproduction problems, and reduce as much as possible the bacterial shedding to the environment as well as the environmental bacterial load. The main tools to achieve these targets are a well-organized and strict vaccination program, as well as reinforced biosecurity measures, especially around parturition. So, let's finish with some basic recommendations for the vets and the farmers. First of all, they should always suspect this disease when they detect signs of the APSW complex. Sampling for laboratory diagnosis and confirmation should take place as soon as possible from the onset of clinical signs. A cornerstone in measures implemented for the prevention of the disease should be a vaccination program that is the only means to limit abortions, improve fertility, and decrease shedding at the same time. Biosecurity is also essential in order to reduce the risk of introduction or reduce the microbial load of the pathogen in the farm level. As it is a zoonotic disease, farmers and vets should protect themselves and the staff through implementation of proper hygiene, proper handling of birth and abortion materials, and the use of PPE, personal protective equipment, especially in the high-risk periods of parturition. And my final recommendation would be to do what you are actually doing right now. Train and educate yourself about the disease, and even more, transfer this knowledge to your clients, your staff, and your families, helping them to protect themselves, but also to contribute to reducing the impact of this important but underestimated disease. And with this, I want to thank you for listening and for your attention.
Thank you very much, George, and thank you for sticking very strictly to the time. That's perfect. I remind you that you can ask any questions now in the chat, and we will deal with them at the end of the third presentation. And now, George, please give the floor to Alda. Alda Natale is also a veterinarian by training. She's working at the Istituto Zooprofilattico Sperimentale di Venezia in Padua, and she's coordinating 3 diagnostic labs there. She's quite familiar with the practical diagnostic aspects of Q fever, and she will prove that to you right now.
Thank you, Vincent. I will talk about practical diagnostic aspects and in particular on which samples should be taken in case of abortion in small ruminants suspected of Q fever or coccidiosis. In practical, we want to be a clinical field guide to post-abortion protocols. We have seen that Q fever, Coxiella, can cause enzootic serial abortion and severe reproductive failure in flocks. Obviously, this causes great economic losses and a severe impact on productivity. We don't have to forget that Q fever in humans can be a severe zoonosis, and the contamination is spread by aerosol and can be also a critical occupational risk. When we face enzootic serial abortions, we have to consider not only Coxiella burnetii, that is a major abortive agent, but we have also to make a differential diagnosis. Depending on the epidemiological situation, on the geographic area or the nation condition, also on the low side, we have to investigate on other possible causes. The most frequent are, for example, chlamydiosis, toxoplasmosis, salmonellosis. Somewhere there is also brucellosis, aspergillosis, and all febrile syndrome. Obviously, we have to consider also non-infectious causes and all the differential diagnoses, because Coxiella can be often found also in mixed infection. We will go through 4 points. The first one is to identify animals to sample, then to select sample types, then we have to choose the right diagnostic test and to interpret results. The first point is to identify animals to be sampled. The gold standard are aborted fetus and placenta, but it is not possible every time to find this kind of samples. So we can concentrate ourselves on the females that had abortion in the last few days. Possible as soon as possible. The best time would be 24-48 hours. If in the lack of this kind of animals, we can concentrate ourselves on females that had reproductive issues in the last 2-3 weeks or more post-lambing or abortion. Optional, we can consider also a control group with females without reproductive issues. Then we have to select the sample types and the sampling hierarchy metrics can have 3 main points. About the main point about the animal to be sampled, the gold standard, we said it is placenta and aborted fetuses. In the lack of this kind of samples, that can be difficult to find, especially in extensive farms, we can consider vaginal swabs. And in the event of a lot of many abortions, we can also consider a pool of 3 samples of vaginal swabs. Then we can also consider complementary serology, and we can consider at least 6 to 10 animals, plus optionally a control group of the same number of sera. About the direct diagnosis, the PCR on bulk tank milk is often considered, but it is a minor interest for diagnosis of abortion. because the positive tank milk proves an active circulation of the agent but cannot ascribe a concurrent abortion due to coccidiosis. So in general, we have no concomitant excretion between vaginal route and milk. So it is very important for epidemiological studies, but not for the diagnosis of clinical problem and abortion. About the diagnostic test, so we have the PCR, that is the main test targeted on the reproductive tract and detect directly the pathogen. And complementary, we have the indirect serology, mainly ELISA test, and the target in this case is the systemic immune response. About the PCR, this is the main test. We have to consider target samples. The cost of this method is medium, and we can reduce the cost by considering pooling samples. It is a direct diagnosis, and the sampling time is time sensitive. As I told before, the best time is as soon as possible, but not more than 7 days for after the abortion event. The transport of the samples must be in a cold chain, and we can also consider the quantitative PCR. This test has a higher cost, but give us more information, because the bacterial load can be directly correlated with the probability of that the abortion event is due really to Coxiella. Environmental contamination are also possible. So if we find a low bacterial load, we can suspect an environmental contamination, or otherwise also a lower excretion that can be possible from asymptomatic animals too. If we consider the pooling of samples, we have to consider that we lose some information. So if we want to use the quantitative PCR, it is better to consider samples taken from each animal. About serology, serology is a complementary test. We can screen a large number of animals. It is a lower cost test. It is an indirect diagnosis, that is mean that we can find also positivity in non-infected animals, because antibodies can persist for a long time after the infection. Furthermore, we can remind that the vaccine cannot distinguish, the serological response cannot distinguish between animal vaccinated and not vaccinated and really infected. So after the use of the vaccine, it is better not to use serology anymore. Finally, how to interpret results. We can follow this simple scheme given by IFSA, and we can start from 2 or more PCR. If all the PCR are positive, we have already confirmed the diagnosis. In case we have 1 positive PCR and 1 negative, we have to follow further investigation. We can look for another animal to sample by PCR, or we can use the serology. If we have 10 serologies with more than 50% positive animals, we can confirm the diagnosis also in this case. On the contrary, we have to follow and go through more investigation. If we can start only by 1 PCR, because we have no other samples available, we can start directly with 10 serologies. As told before, the association between the PCR results and serology results can confirm, exclude, or remain in a gray zone that require further investigation. About the interpretation of serology, we have to recall that also that shedding animals can be still or remain seronegative. But in general, the highly positive animals or simply seropositive are generally the best interesting ones. So in general, high shedder or chronic shedder are seropositive. The aborting female in the acute event may be still seronegative at the time of abortion, so it could be a good idea to test again this animal after 2-3 weeks to confirm the diagnosis. And the within herd prevalence is very important because in a group of animals that had the circulation of the agent, seroprevalence is high, so it is important to estimate the prevalence. Finally, we can have asymptomatic animals that can excrete during normal parturition. To final recommendation, we can recall again the 4 main points. So the first point is to identify animals to be sampled, so animals with reproductive issues. Then select sample types, placenta, fetus, vaginal swabs, and sera as complementary test. We have to choose the diagnostic test that can be a combination of direct PCR, real-time PCR, or quantitative PCR, and serology, generally represented by ELISA test. And finally, to interpret results by EFSA protocols.
Thank you so much, Alda. Thanks to the audience. We can still have questions in the chat, and before that, I give the floor to Piet Vellema. Piet is from the Netherlands. He's also a vet by training. He's a diplomate of the European College of Small Ruminant Health Management, so that means he's a specialist in small ruminant health, and he will address the One Health aspect of Q fever in his presentation. Piet, the floor is yours.
Thank you, Vincent. Dear audience, for the next 15 minutes I'm going to give you a short overview of the Q fever outbreak in the Netherlands between 2007 and 2010, and give some background information on the animal part of the outbreak and also on the zoonotic aspects of this outbreak. Going back to the history of Q fever in the Netherlands, Q fever was diagnosed for the first time in the Netherlands in 1956. The strange thing is there has been quite some screening in the years before 1956, and no cases were found then. And in this year, 3 different persons were found to have Q fever. No connection between them, no sources found. But that was the start of where the Netherlands were involved in Q fever. In 1978, Q fever became a notifiable disease in humans. And if you look at the average number of cases in humans between the year of the disease becoming notifiable and the outbreak starting in 2007, the average number of cases was only 17 per annum. And if you look at the number of hospitalized persons, it's about 49 between 1994 and 2001. And then at a sudden point in 2007, we saw a rise in human Q fever cases. And this is the picture, a short description of what I've described before, between, at the left, 1997 and the start of the outbreak in 2007. An average of only 17 cases per annum, and then a sudden rise with about 200 cases in 2007. If we go back to 2005, 2 years before the first cases of the first human cases were found, Coxiella burnetii was diagnosed for the first time as a cause of abortion on a dairy goat farm in the southern part of the Netherlands. We didn't have any experience with that disease, so what we did is we notified the farming industry in both the Ministry of Agriculture and the Ministry of Health that we had found Coxiella as a cause of abortion, and looking from what has been described in literature, we warned for a possible outbreak of Q fever in the rural population about the farms where the abortion had taken place. So that was 2005, and a second case was also found in the same year on a dairy goat farm in the same region. Going back to the years to follow, so 2005, 2 dairy goat farms with an abortion storm. 2006, 6 dairy goat farms and 1 dairy sheep farm, and there was a big difference between the dairy sheep farms and the dairy goat farms, because the problem on those dairy sheep farms was much a minor problem compared to what we saw in dairy goat farms. 2007, 7 dairy goat farms, and then the first human cases were notified and confirmed. In 2008, 1 dairy sheep farm and 7 dairy goat farms, and in 2009, 6 dairy goat farms. Abortion rates on those infected dairy goat farms were between 10 and 60%. And if you have an impression of the dairy goat industry in the Netherlands, the average number of goats in that period was around 1,000 animals per farm. It has gone up to more than 1,400 animals per farm at this moment. But if you imagine that 10 to 60% doesn't imply all the animals on the farm, because on most of these farms you have what we call an extended lactation, and only parts of these animals are pregnant at the same time. So this is the situation before 2005, no cases ever confirmed, and as a consequence of all the measures I come to later on, after 2009 we never had any confirmed cases of abortions caused by Coxiella on dairy sheep and dairy goat farms. Then, as a sudden outbreak in 2007, we had an outbreak of Q fever in the area in and around the village of Hurpen, where we found a cluster of 55 patients, and we looked at the background of them with people from the veterinary field and the human fields to get an impression what was going on. And then we saw something like an increased risk for smokers and those in contact what was being meant as contact with hay and manure, so contact with probably farming industry. We did quite a seroprevalence study in the village of Hurpen, and we found that 24% of the inhabitants of that village had recently seroconverted. So again, the same picture with a bit more information. At the left again, 1997, and then a sudden rise to almost 200 Q fever patients in 2007, and you see the area where the patients did come from, and the larger the spot, the more human cases. And I come in later slides to another way of showing how the infection looks like. So the darker brown the area, the more human cases found. And this is the same picture as this one in another way. And for the years 2008, 2009, 2010, I have the same picture. The darker area, the darker brown the area, the larger the human cases found. So this is 2007, almost 200 confirmed cases of Q fever. Then you get 2008, and the number of confirmed human cases were almost 1,000. And then in 2009, the number of confirmed human cases were 2,300. Then at the left, the same picture of 2009, the red area, the area closed in with a red line is the province of what we call Noord-Brabant. So there was the center of the Q fever outbreak. And if you look at the right picture, you see the darker blue the area, the higher the number of dairy goats in the same area. And if you look at the map, you see different spots with different colors indicating the year in which abortion outbreaks have taken place. And if you compare these two maps, you see that the outbreaks, abortion outbreaks and the human patients were more or less located in the same area. Not everywhere. If you look at the outbreak in the northern part, no human cases were almost found there, and in the eastern part the same applies. Although the farm in the half midway in the Netherlands in the eastern part, that was a dairy goat farm where movement of animals from an infected farm in the southern part of the Netherlands had taken place. So this is an overview, and there's always then a question, were dairy goats the cause of the outbreak? All the information we gathered make it plausible that dairy goats mainly were the cause of the outbreak of Q fever. If you then look at what has been done about notified and confirmed human cases and background information gathered from these patients, most of them, and you see these 1,009 patients and 621 of them were diagnosed with pneumonia, is 62% of all those cases. Hepatitis is also quite often mentioned as a clinical sign of Q fever, but in the Netherlands this percentage was below 1%. Endocarditis is about 3%, and several others were also part of this clinical spectrum. In another way indicated, if you look at the clinical signs at the left side and the number of patients where these clinical signs were found, and the percentage between brackets, fever, more than 90%, muscle pain, around 60%, headache. It's in the earlier years of the Australian Q fever outbreak, headache was mentioned as a main clinical sign. You see the back also in this overview, around 70% of them had headache, several complaints of malaise, 60%, then pneumonia, like problems like coughing, shortness of brain, around 60%, and then at the end of it, fatigue, almost 80% of them showing this clinical sign. What measures? That was quite difficult at the start because we didn't have experience. In June 2008, 1 year after the first outbreak, Q fever was made a notifiable disease for dairy sheep and dairy goats. Quite some hygiene measures were taken without we knowing what the possible results of that would be. We started vaccination in October 2008, aiming at reducing infection pressure, but we used Coxsavax then and had a problem with the authorities at that moment because it hadn't been registered in the Netherlands at that moment. So on the one hand, with the information available, we'd like to start using it, and on the other hand, we had a problem with the registration. So we started vaccination in October 2008 on a voluntary base in the area where we had found most of the human Q fever patients. Then in 2009, it was compulsory in the province of North Brabant, in the picture I showed you earlier, covered with a red line. And from 2010 onwards, and it still is compulsory on a yearly basis for all dairy sheep and dairy goats in the Netherlands, also for small ruminants on children's farms, social care farms and zoos, and on small ruminant farms where visitors are admitted during the lambing period to the stable. What we also did to follow what was going on in the field, from October 2009 onwards, we started monitoring bulk milking samples using a PCR. It started in October 2009 and it's still going on. And you see the left bar on this picture is September, October 2009, almost 24% of the herds and flocks being PCR positive. It went down after vaccination on a dramatic way, and since 2016 we have never found PCR positive bulk milk tank results since then. So this is an overview of what you saw. Until 2007, only 17 cases per annum. Then 2007, 200 cases. 2008, almost 1,000. 2009, 2,300. Going back to about 400 after all the measures we have taken in 2010. And since then, the infection just faded out. And if you look at the website of the RIVM, where Q fever is mentioned in this way, you see the number of confirmed cases in 2013, only 19, in 2023, 6, in 2024, only 1. So the average number before the outbreak was 17, and it went down afterwards to 15. Although a lot of attention has been paid about what was going on, a lot more measures of a lot more investigations on humans with clinical science performed, and still the number is very, very low. A short overview of what's going on now. Bulk tank milk monitoring using a PCR still takes place on every dairy sheep and dairy goat farm every month. The sheep and goats on these farms and the farms I mentioned earlier have to be vaccinated annually, and we think that all the measures we have taken, that vaccination has played a key role in controlling the Dutch Q fever outbreak, and we could easily follow what was going on by the bulk tank milk monitoring that is still in place. So that's my short description of what took place in the Netherlands in the years between 2007 and 2010.
Thank you so much, Piet, for sharing that with us. And thank you for respecting the time frame we've had. This is perfect on time. I don't have questions so far in the chat, but I prepared some, of course, in that case. And I'll start with Alda, because Alda, you said that you have to choose a test when you're facing an outbreak of abortions, for instance. So what is your recommendation to a vet who wasn't aware of or wasn't thinking of Q fever before he attended our webinar, and now he's having a small idea in his head. He wants to, next abortion storm in a herd, he wants to test for Q fever. What do you advise him?
The first recommendation is to contact the local laboratory, because every laboratory has slightly different rules. Some laboratory prefer vaginal swabs or swabs in general. Some other prefer the entire fetus or the placenta. So the best first step is to contact the laboratory. Then it is important to perform the systematic surveillance on abortion in general. So not only for Q fever, but for all the causes, the possible causes and differential diagnosis, because we can have co-infection and some other important infection.
Okay. And when you mentioned that on quantitative PCR, when you have a low level of genetic load, you suspect environmental contamination of the samples. Are you faced with this problem often in the laboratory?
Yes, it is possible. Into the laboratory, it is difficult to distinguish between the environmental contamination or a low amount of shedding. The best practice is to clean very well the part if we are taking vaginal swabs, and in case of collecting placenta from the ground, obviously the environmental contamination is possible. The quantitative PCR can give us the suspect of environmental contamination, while in case we have a big amount of DNA of Coxiella, we can be almost sure that the event of abortion is linked to Coxiella.
And that's the thresholds you showed on the figure with the meter.
Yes, the 10 to the 4 is a high bacterial load.
Okay, thank you so much. George, you also mentioned environmental contamination and the importance of hygiene, but removing fetuses and placenta, is it enough to limit environmental contamination?
No, it's not enough. Unfortunately, as I showed you in the data, more or less almost every farm, at least in many countries, seems to be exposed to the bacterium. Of course, it's important to try to reduce as much as you can the bacterial load in the environment with disinfection programs, with hygiene, with all the biosecurity measures, even with testing the introduction of the new animals. But we understand that the animals play a really important role in with their shedding of the bacterium. So this must be combined, as we saw in the example in the Netherlands. It's an obvious example how shedding can be reduced with the vaccination program that should be implemented at the same time with all the biosecurity and disinfection programs.
So that's more a holistic view than focus.
You need a combination of two.
And for the farmer that grabs the placenta, do you have a specific recommendation?
You know, there are many zoonotic diseases. One of them is Coxiella, there is also Brucella. There are so many zoonotic pathogens that he must be very careful with dealing with all these materials. Parturition, even we saw that even healthy animals will shed the bacterium, the abortion materials of course as well. So he needs to use all the time personal protective equipment, to remove all these, try to destroy them with sanitary burial or burning, and reduce with these ways the bacterial load in the environment as much as he can.
Okay. And if the farm hasn't got a known status, do you consider that it should be a reason for increasing the hygienic measures and the protection measures? Of course,
of course. Even the hygiene measures, all the measures will play their role in even the introduction of the pathogen in the farm. Even if he in a negative farm, in countries that we know that there is a huge exposure of the farms, even the introduction can be avoided, the possibilities be reduced by doing all these hygiene measures and doing the disinfections, the programs, the vaccination, the disinfection, all these play their role protecting his farm and at the same time protecting him as well. It's really important for this pathogen. Him as well, the staff, his family.
Okay. Yeah, that's main. And elaborating also on the hygienic measures, Piet, you just mentioned them on your slide, but can you go further into details of the hygienic measures that were implemented at the time of the outbreaks?
Yeah, we've had quite some sessions, because what I explained is at the start we didn't have any experience.
That's
what
right.
we are dealing with, and then we are thinking about how to get rid of placentas and dead fetuses. So we explained farmers' vets to collect them and render them. So they were collected for rendering, while normally placentas are not collected for rendering. So that was one of the main things. We've also had quite some discussions about the possible role of manure and spreading manure on the field and the possible risk of that. And what we did then is look what was going on when we got the manure from the stable, heaped it, and we have studied what's going on in the manure heap. And what we see then, if you don't cover a manure heap, the temperature rises in 1, 2, 3 days to about 70, 73 degrees. So that means that in the outer layer of a manure heap, not covered, all Coxiellas will just die out, not in the center of it. So what you have to do is you treat the manure this way, turn it a while, and then almost no risk for manure is there anymore. So on the one hand, we advised be very careful with manure. On the other hand, when we had done this trial, we came to a completely different opinion. The government started to say, you have to cover your manure with plastics, but plastics make it impossible for oxygen to go in, and oxygen is essential for getting a rise in temperature. So at the start, we came up with hygiene measures without knowing the possible results of it.
Okay.
And I think the collecting of placentas in these fetuses for rendering were the main thing, and after the trial with the manure, we knew more or less of the background of that. But quite some other measures, normal hygiene measures, I don't think there is any influence on Coxiella dissemination.
Okay. One other puzzling thing, apart from the real success of the vaccination of the animals regarding the human contamination, because we see a very sharp drop, we still see a tail of cases being regularly, not a large number of them, but being regularly diagnosed. Can you comment on that also?
Yes, I've tried to do it by showing the picture. Before the outbreak, we had on average 17 cases per annum, and after 2011 or 13, it was only 15, with a lot more attention, with a lot more people tested. So we're back now to a lower level than we started. That's the one thing. If you look at a farm where abortion has taken place, as soon as you have vaccinated for the second time, excretion in the bulk milk tank goes to almost zero. But what happens with a certain delay is it going to rise again. And if you follow individual animals on a farm with a history of abortion, you see that animals that have had an infection before their first kidding without being infected can shed Coxiella again later on for more than 1 or 2 years.
Mm-hmm.
So there is still some shedding on those farms, and I think that combination of what happens on the farm, where not every animal that has been vaccinated before the first pregnancy can be sometime a smaller risk. But if you look at the total figure, it's the drop in cases, and you have to imagine we started vaccination in the center of it, 2008, then in 2009 the province of North Brabant, and then only from 2010 onwards the rest of the country. So there is a certain delay, and it's not because we didn't want to, just because we couldn't get the number of vaccines we needed.
Okay. So that was a progressive implementation,
yes,
but with a strong success when we look back from now.
Yeah.
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 Okay. And one of the missions of the European Q Fever Committee is to raise awareness on Q fever. But can you also comment on the impact this outbreak has had on the awareness on Q fever in the Netherlands? Not, of course, in the hotspot of the animal outbreaks, but among the farmer community at a wider scale.
Yes, that's quite difficult, even in a country where it was decided at the end of 2009 to kill all pregnant animals on PCR positive farms. In that area where that took place, a high percentage of the population knew what was going on. But the Netherlands is only a small country, 200 by 300 kilometers about. But in the northern half of the country, they argued saying it's a southern problem, it's not our problem.
Okay.
Yeah. Mm-hmm. So and even if you look where television made announcements of goats being killed, it's been shown on television quite often. Even if you look at what you know about the knowledge of human doctors in the northern part of the country, there were many of them didn't know what Q fever was. So communication of a disease is not so easy.
Okay. That's a good thing for our own work, because we know there is still lots of work to do on awareness about Q fever.
Yeah.
I have one question that happened to arrive on the chat, and it's also for you, Piet. Could the difference between incidence pre and post-outbreak be partially justified by underdiagnosis, low health-seeking behavior pre-outbreak, and higher health-seeking behavior post-outbreak?
Yeah, if you look and if you look back, you never know what you knew and what you missed. So if you look at the situation between 1978 and the outbreak, where it was a notifiable disease, I think it's normally to think that quite some clinical cases could have been missed. That's still the situation after the outbreak, but there is so much attention in the human and veterinary field that I think with the attention paid to it, the large number of people tested, I think the information and the overview we have from the situation now is much better, gives you much more information than before the
So more accurate.
Yes, I think so.
Yeah, okay. And George, you have the same experience? Because in Greece you've had not such a wide number of cases and a wild outbreak, but you have had some outbreaks also with human cases.
Yeah, exactly.
Has it risen awareness locally, or?
Unfortunately, it seems that human doctors do not undervalue this disease. Okay. Usually they look at it like a flu-like syndrome, and in Greece especially, that the overuse of antibiotics is really extensive. Giving some antibiotics, they think that it's okay, and they just declare this as a flu-like syndrome, and they do not proceed to confirmatory diagnosis. But at the same time, we see, and our research shows this, and our publications, we have huge exposure of the animals to Coxiella. We have large rates of abortions due to Coxiella, and other colleagues' works shows the last years huge exposure of humans as well, and especially in the farming communities, in the villages. So we all know that it's really underdiagnosed in the hospital level.
Okay.
Right now in our country.
Okay. And Alda, same question for you regarding awareness. Do you think an outbreak increases for a long time the awareness among vets or GPs, for instance?
Yes, historically we had some little outbreaks in Italy, limited to 100 people maximum. Very recently we had a little outbreak of 4 people hospitalized, linked to a parturition of a single cow in a little farm open to tourists.
Okay. So
in general, we had some single cases detected by GP practitioners, and we help sometimes with the diagnosis with our laboratory.
okay, yeah, cool. Thank you. So we are nearing the end of this session. I want to remind you that most of the graphs that were presented by Alda and also by George are from the fact sheet that the European Committee is producing, and that are freely available at the moment in English, but later on in other languages on the dedicated website, and that you can register to our website to receive updated news regularly. And with this, and with no further question in the chat, I would like to close this webinar, and thank you very much. You've been a beautiful audience. Thanks a lot.